Hi guys, I've been told that I need to speed things up, so I'm going to try and do that this time. As you can tell, in this video we'll be going through the nephron and how its structures are responsible for ultimately the excretion of urea while retaining water, glucose and amino acids through selective reabsorption. Okay, so there's three stages that we need to know. The first one is called ultrafiltration, and we'll do that first. Ultrafiltration. Okay, so I'm going to be focusing on the main concepts here. Please forgive me if I might miss a fact or two from the textbook, but I think with something that can get quite detailed, often we lose what's really going on here, and ultimately that's what the exam will test, whether you have a broad appreciation of what's going on here. The details you'll fill in when you do your revision. Okay, right, so ultrafiltration, it happens here, the glomerulus here, and the Bowman's capsule right there. My diagram's a little bit dodgy, but I've tried to adapt it so I can go through all the three stages on one diagram. Okay, so ultrafiltration. So the route that the blood will be taking, and so blood's going to come in here. That's the glomerulus right there. It's going to come out here, loop around in these blood vessels that I've shown here. Okay, and then come back and rejoin the vein. What I'll do is I'll show the route of the blood in red. Okay, so come in there. That's the afferent arteriole, right? Oxygenated blood right there. Okay, and the afferent arteriole has a larger diameter than the efferent arteriole. Efferent arteriole, and because it has a larger diameter here than here, there's an increase in pressure in these capillaries. Okay, all right. So ultrafiltration reducing blood vessel diameter. Okay, so there's a reducing blood vessel diameter which increases the hydrostatic pressure. And essentially it's because of this increase in hydrostatic pressure that you get stuff forced out of the blood that's moving in these capillaries. Okay, so things are forced out of the capillary into the Bowman's capsule. Okay, so that's the main idea here, that because of this capillary, or this arteriole having a wider diameter than that one, there's increasing pressure, and that increasing pressure, hydrostatic pressure, forces things out. Now, when the things move out, they have to move across three barriers. Okay, so three barriers. Okay, the first one is the capillary wall, which has gaps, right? So the capillary wall, the capillary walls have gaps between them, so things can move out. The second one is the most important one, and this is the mesh, or the basement membrane, which is formed of a mesh of collagen, fibers, and glycoproteins. Okay, and because this is really fine, so it only allows small things through. And again, finally, we have the third barrier, which is formed of the cells forming the Bowman's capsule. Okay, now these cells have processes which kind of interlink with each other, but there's gaps between those processes, and between those gaps, things can move through. Okay, so I'll put it here: gaps between podocytes. So podocytes is the name of these cells. Gaps between podocytes processes. Okay, so finger-like protrusions type things, and they link together like this, but there's gaps, so things move through. Okay, so as a result of these three barriers, things which are less than 69,000 relative molecular mass units will be forced out of the blood and into the Bowman's capsule. Okay, so they'll end up here in what is called the glomerular filtrate. Right, so anything that ends up in here forms the glomerular filtrate. Okay, glomerular filtrate. And the things that we will have are all the molecules that are smaller than this size. Okay, so we are thinking about water being forced out. We are thinking about inorganic ions, sodium, potassium, chloride ions. Yeah, they'll be forced out. Water, inorganic ions, urea. That's the thing that we don't want anymore. We will have glucose in there. We'll have amino acids in there, and we'll have small proteins. Okay, any proteins which happen to be less than that size. Okay, is this all from there? Right. Okay, so this is important to remember, that all this stuff ends up in here. Okay, now this is the key point here, that of all the things that are moving out of the blood at this point, only urea is really the thing that we don't want anymore in the blood. Everything else is rather important. So the rest of what happens here, the rest of what happens in the nephron, i.e. in the proximal convoluted tubule right here, and in the loop of Henle, or due to the loop of Henle, that's responsible to get everything back. The water, the glucose, amino acids, that all needs to be taken back into the blood, okay? Leaving then finally urea, which we don't want back. That remains in the filtrate, forming the urine, which eventually ends up in the collecting duct, onto the ureter, bladder, and then out. Yeah? So that will be then excreted. So of all these things that come out, only urea is the thing that we don't want anymore. Okay? So everything else we got to get back, and that's really what our discussion is going to be about. How does all that stuff make it back into the blood? Okay? So all these important things are now in the glomerular filtrate. We've got glucose, we've got urea, we've got sodium ions, we've got chloride ions, we've got amino acids. Okay, I'm going to ignore the proteins bit. So then we need to talk about how those things get back in the blood, except for, remember, urea. We don't want this back in the blood. Hopefully that's going to stay in the system, forms the urine and exits the body. Okay? Right, so the next stage then, this glomerular filtrate is going to move through the proximal convoluted tubule. Okay, now please excuse my diagram, I've tried to show the structure of the cells forming the tubule. They have microvilli to increase their surface area to help the process of selective reabsorption, and I think I'll do that here. Okay, so first we did ultrafiltration, next we're going to look at selective reabsorption. Okay, and remember that's happening in the proximal convoluted tubule. Right? Convoluted, why? Why does it have so many twists and turns? Well, the longer that tube is, I guess the greater surface area we have to reabsorb all this. We need to reabsorb. So, what gets reabsorbed in the proximal convoluted tubule? Water. Well, let's not start with water. First, glucose, amino acids, and as a result of that, water will follow by osmosis. So how is it that we get glucose and amino acids? Remember, they exited the blood here. We need to get them back in the blood here, right? So the blood went through here, came back out here, went there. It's now flowing through here, okay? And now all of the useful stuff that came out here, we need to put it back in there. How do we do that? I'll tell you. So, we've got all this stuff here. Now, the main player in this process, right, so the main player here is a sodium-potassium pump. And it will use the energy from ATP breakdown to ADP plus a phosphate group there. Okay, that's why you got mitochondria there. But it's going to use the energy from the breakdown of that to move sodium ions against a concentration gradient. So sodium ions it will pump out of the cell, while it will move potassium ions in. So I'll just put that down. Sodium ions pumped out, potassium ions in. Okay. Now, what that will do is it will increase the concentration of sodium outside the cell, but it will reduce the concentration of the sodium inside the cell. Okay. Now, remember, one of the things that we've got in this glomerular filtrate is sodium, because it came out of the blood. Right, as well as that, glucose and amino acids and urea. Okay, anything else we missed? Yep, okay. So, what happens is that active transport, and this is important, right? So, active transport of sodium out of cells. All right, so sodium is pumped out. Now what that means is that sodium concentration is reduced inside, so sodium, there's a concentration gradient now. Sodium is low here, but high here. So therefore, sodium is going to diffuse from there into this cell. But this is where it gets clever, because there's a co-transporter in that cell which it co-transports sodium down its concentration gradient at the same time transporting a glucose in as well. Okay? And the same process is done with other co-transporters that transport sodium in with an amino acid. Okay? So, we're creating a gradient whereby sodium will diffuse into the cell, but because it's a co-transporter, it will transport sodium and glucose at the same time. Right? That transporter won't be able to transport sodium on its own. It doesn't work like that. It will only work when both a sodium and a glucose click into the protein on one side they both get transported down sodium or down the concentration gradient of sodium. Okay, so glucose comes in as a result. So now that the glucose's concentration is increasing here within the cell, it will also then diffuse out. Okay, so glucose then diffuses out of the cell because its concentration has increased within this cell. And when its concentration increases here in this space outside this cell, it will then diffuse into the capillary. Yeah, because its concentration in the capillary in the blood is now low because of what happened at ultrafiltration. It came out. And because it's got a low concentration in the blood, it will diffuse from the extracellular space into the capillary, into the blood. Okay? And remember, the same process will apply to amino acid. So this cell will be pumping out sodium. Okay? The sodium concentration in this cell will be low as a result of that. There'll be a co-transporter that transports sodium in at the same time as an amino acid. The amino acid concentration inside the cell will therefore increase. The amino acids will therefore diffuse out of the cell, and once the amino acid concentration increases outside this cell, it will then diffuse into the capillary, because again, the capillary, the blood concentration of amino acids will be low. Okay, so what I'll do is I'm going to use a dotted line for any diffusion that's happening. Okay. And anyway, I'll put the line like this for the co-transporter. For any diffusion, I'll put a dotted line. Okay. Remember, any diffusion that's happening across a membrane for polar molecules, it will need a protein to facilitate that. Okay, so there's three types of diffusion going on, so three types of movement. The initial movement, pumping out, active transport of the sodium out of the cell, followed by the facilitated diffusion by the co-transporter of the sodium and the glucose into the cell. Once the glucose increases in the cell, it's going to be moving by facilitated diffusion out of that cell into this space, but then it will move by regular diffusion from this space into the capillary through the gaps in the capillary wall. Okay, so active transport of sodium out of cells. Sodium and glucose, or sodium and amino acids move into cell from the glomerular filtrate by diffusion. And I'll just mention the co-transporter here, that's important. The co-transporter, all right? And then glucose and amino acids diffuse out of cell and into the blood. Okay, right? It's looking a bit busy, but hopefully it has made sense. Okay, remember, pumping sodium out is the first step. You lower the sodium concentration, sodium and glucose via the co-transporter into the cell, increase their concentration in the cell, and they diffuse out of the cell. Remember, because their concentrations are going to be comparatively high in this place, they're not going to want to diffuse that way. They will be diffusing where their concentration is lower, i.e. in the tissue. And from that tissue, they will be diffusing where their concentration is even lower, i.e. in the blood. Okay? So make sure that you can explain the movement of these things. If it doesn't make sense, you have to figure out why these things are happening. If it's not making sense, you won't be able to remember it. Okay, so in this way, by the time things get here, the the amino acids and glucose have moved in. Now, this is the other interesting bit. Now, because the glucose and amino acids are being moved out from this space into here and into the cell here, and then from the cell into the blood capillary, we are lowering the water potential of first the cell, then the tissue fluid, and then the blood itself. We are lowering that water potential because we're moving solutes away from the glomerular filtrate into the cell, into the tissue fluid, into the capillary, and therefore water is going to follow by osmosis. So, I forgot to mention that water's here, right? A lot of water, the majority of water, will actually move out by osmosis, following the movement of the sodium, the glucose, the amino acids. As these things move and lower the water potential in other places, water follows, ultimately back into the blood. So where we are, guys, where we are now is at a stage where lots of stuff was forced out of the blood in the ultrafiltration step, but because of selective reabsorption, we've got the amino acids, glucose back, and a lot of the water. What we have to think about now is getting the remaining water back and remembering that we've still got sodium and chloride ions there, but also we've got urea here. Okay, so the real question is, how do we get the remaining water back using the sodium and chloride ions, and leave the urea at where it is in this fluid? We don't want to get that back. That's the next step. Okay, so this is now the loop of Henle. Okay, and it's got a descending limb, it's got an ascending limb. There's a distal convoluted tubule following that, and that's the collecting duct there. So what we want to do now is look at how the remaining water is going to be reabsorbed. Okay, now, and this is really the function of the loop of Henle, is the reabsorption of water. All right, now this process that we talked about in the proximal convoluted tubule, yes, it led to a lot of the water moving back into the blood. However, we didn't lower the water potential enough to draw out all of the water by osmosis. A lot of it, much of it is still there. Most of it did go back, but there's still quite a bit there, and we need to get that back in the blood. How does that happen? Well, the main function of the loop of Henle is really to lower the water potential as much as possible to draw out the water from this tubule of the loop of Henle. So if the water potential outside really is low enough, the water will move out by osmosis, hopefully then into the capillary. Okay? Now, what I'm going to do is just skip this first limb here. We'll come back to that, I'm not going to ignore it entirely. But the really interesting stuff is happening here in the ascending limb, right? And basically, as a result of what happens here, stuff happens here, which is important, okay? So, what I'll do here is talk about reabsorption of water, loop of Henle. Okay, so that's the job of the loop of Henle, that remaining water, getting it back. How do we do that? Well, the fluid is going to move through the descending limb and then up to the down through the descending limb and then up through the ascending limb. Now the ascending limb does something interesting. It actively transports, so I'm going to make, remember there's going to be cells here that are responsible for this. I'm kind of simplifying it by showing it this way, but really it's happening in the cells which form the tubule, okay? So the ATP is broken down to ADP, releasing energy, remember? Okay? And that energy is used to pump out, remember there's sodium and chloride ions in here. So sodium and chloride are moved out into this extracellular space. Okay? So chloride, sodium, right? So ATP, okay, broken down. ATP is broken down, sodium and chloride pumped out of this fluid. Remember, those ions are in this fluid and they can be pumped out. As a result, we lower the water potential in this area. Okay, so this is the medulla that we're looking at here. The water potential is lowered here because we're pumping out solutes, we're pumping out inorganic ions. Water potential reduces here, and this causes whatever, water here causes it to move out by osmosis. Okay, so water is moving out of this descending limb because the surrounding area has got such a low water potential. And the surrounding area has a low water potential because of the action of the ascending limb pumping out sodium and chloride ions into this space has caused the water potential to be so low, water moves out. Okay? Now, the water moves out into the extracellular space, into the tissue fluid, and from the tissue fluid, it will move into the capillaries. Okay, so once it goes out into the tissue fluid, it will move into the capillaries, and it has then rejoined the blood. Circulation. All right. Now, that's the main point of the Loop of Henle. I mean, there are details that we could discuss, but I think this is the main point. Interestingly, there might be some confusion about what happens to the sodium and chloride ions. They do rejoin the blood at some stage, but the important thing to know is that the sodium and chloride ions cannot move into here. That would be counterproductive, right? So the sodium and chloride move into this space right here, lower the water potential, draw the water out by osmosis from the descending limb, and from there the water moves into the capillaries, and there we have it. Then, so the sodium and chloride are gone now from there. The water now reabsorbed, yep, and water will even be drawn out from the collecting duct. So if there's any water still in, I mean, there's always going to be water there, okay? But if need be, water can be drawn out even from the collecting duct. Okay? So once we get water outside of these tubes, right? Once we get water into the extracellular space, into the tissue fluid, it will then move by osmosis into the capillary. Okay? Right, and that's the reabsorption of water. So what were the main points? Reabsorption of water. Essentially, active transport of sodium and chloride ions out of ascending limb into the medulla, basically. I mean, this is the medulla, right? So into the medulla. Okay. Let's just call that tissue fluid because it's between the cells, not within the cells anymore. Okay, so into the tissue fluid, lowers medulla water potential, water moves out from descending, from the descending limb. Okay, water moves out from descending limb. Remember, we must always mention osmosis, diffusion, active transport. These are important things. So it's moving out as a result by osmosis and then into capillary. Okay, and there we have it. There are other details regarding this, you know, solute concentration, the permeabilities of that compared to that, but I think if you understand this, then you know you can fit the details in later. This is the main thing from my point of view. Okay, guys. Any questions, comments, feedback? Greatly appreciated. Good luck.